BPDCN is a rare subtype of leukemia for which we have a center at Dana-Farber that studies the disease in the laboratory and in clinical trials for patients. And there's only one approved drug for BPDCN, and it's called tagraxofusp. It targets a molecule called CD123 on the surface of the BPDCN cells. And while tagraxofusp is an efficacious drug and it helps get many patients into remission, there are some challenges with tagraxofusp, being that we have to give daily infusions intravenously for 5 days, and that we also have to admit patients to the hospital during the first cycle to monitor for side effects of therapy. So with that basis and some work that we had done in the laboratory trying to understand mechanisms of response and resistance to tagraxofusp in BPDCN, where we found that the resistance to tagraxofusp can be overcome by treatment with a drug called azacitidine, which is a chemotherapy drug that we already use in leukemia, and that venetoclax, which is a BCL2 inhibitor, is particularly potent in BPDCN cells. So with that information, we first performed a study in patients with AML and MDS that was published last year, where we figured out the safe doses to combine the triplet regimen of tagraxofusp, azacitidine, and venetoclax. And here what we are presenting is the phase 2 data of 27 patients, about half and half each with previously untreated, so newly diagnosed BPDCN, or relapsed BPDCN, where we use the triplet combination. And the bottom line is that the combination was safe. We didn't see any new safety signals that were unexpected based on what we know about tagraxofusp alone or venetoclax azacitidine alone, or from the triplet that we had seen in patients with AML. And secondly, we saw a good signal of efficacy. So we saw a very high response rate, including that most patients achieved a complete response, including nearly all of the patients with previously untreated disease, and that we were able to get many patients to an allogeneic stem cell transplant after the treatment, which is usually our goal with patients with BPDCN after we get them into remission. And it looked like the capillary leak syndrome, which is the key toxicity of tagraxofusp that we follow patients for in the hospital, might be the same or even a little bit better with this regimen of tagraxofusp, venetoclax, and azacitidine, probably because in this regimen we give only 3 days of tagraxofusp as opposed to the 5 days when it's given as a single agent in BPDCN. So we're really excited about these results, and we think that this could actually lead to a new standard of care treatment for BPDCN. A caveat is that, of course, this regimen was not tested head-to-head against any of the other regimens we use in BPDCN. It's very difficult to do that in this rare disease. But I think given that there's a lot of familiarity with venetoclax and azacitidine already in leukemia patients, it's an approved regimen for patients with AML. And because we're combining it with an already approved drug in BPDCN, we hope that this may become a new regimen that can help more patients with potentially less toxicity.